So I'll be talking about waves and how waves appear in all kinds of unexpected contexts across nature and society. Okay, so when you hear the word waves, what picture comes to your mind? Sorry? Uh, a little louder? I can't. Sinusoidal waves, then? Just a curve. Okay, anything else? Sorry? Sea beach. Fantastic. Okay, something like this. Okay, so this is actually a video. Let me see if this thing. Oh, sorry. Uh, let me see if it runs. Yeah. So this in this video you can see that there's a ball which is kind of floating on the ocean. Now you can see there is things moving, right? The waves are coming forward, but the ball seems to be pretty much where it is. Okay. So my next question to you is, what's moving? So again, let me go back to this video. Okay. So you can see, like, sorry, yikes. So if you focus on, let's say this one, you can see that, it's traveling from there to here and then disappears off screen. But the ball doesn't seem to be moving at all, right? So what's moving? It's the disturbance that's moving. Fantastic, right? And so if you look at a schematic view of what a wave is, essentially each part of the ocean, you can think of it as that there is a material flow occurring, but these are circular currents. So the water particle itself is not actually traveling from here to there. Okay, there is a very small localized circular current which is causing it to circulate like this. Okay, but as this moves, it causes its neighbor to move at a phase. I hope phase is a concept which is familiar to all of you. So it's basically, if I look at a particular point on this circular rotor, I ask, okay, I tag a particular particle, okay, and I ask, okay, where is this particular tagged particle at when I'm looking at it at different points of time. So let's say I tag a particle over here. So as I look at it, I will see that it will basically go round and round and round, okay, with a particular frequency. All right. And the neighboring point would also go like this, but as you can see, its phase is slightly behind, and then the neighboring circular rotor, its phase is slightly behind, and so on. So what is happening is that this one has the, like the water particle which is actually at the top of this column, its height is located over here. The water particle which is at the top of this column, its height is here, and the water particle which is at the top of this rotor, its height is here, and so on. So if you trace a line connecting the peak of each of these rotors, or if you were the instantaneous position of each of these rotors, you will actually see something like this sinusoidal curve. So someone said sinusoids. So that's also correct, because if you look at this shape, it actually has a sinusoidal form. Okay. So what we see from here is that a wave is very intimately connected with the concept of oscillators. Okay. Now what's an oscillator? Something which has a period. Okay, so it could be, for example, a pendulum. Okay, it could be a swing. Okay, so in a pendulum, for example, we would see the periodic motion as swinging back and forth to some maximal amplitude and then going back. Okay, so it will trace something like a sinusoid. Right. Now, from the perspective of dynamical systems theory, we tend to view this slightly differently. Okay, so we tend to see it in what is known as a phase space. Okay, so for those of you who have taken some elementary physics, you'd recall that if you have a particle on a spring such that it's bobbing up and down, you can say that the force of the spring, which is causing it to spring back, that restoring force is proportional to the displacement of this particle from its equilibrium position. So if we call that x, the restoring force actually works against the extension, so it's negative of, it's proportional to negative x, and the proportionality constant is k. So it's basically the spring constant. It tells you how tight the spring is, right? So we say restoring force equal to minus kx. Now, recall your Newton's laws. So Newton's second law says that force is equal to mass times acceleration. So we know that the acceleration can be written as the second derivative of the displacement, right? So that times mass will give you the restoring force, which is just minus kx. So basically we end up with a second order differential equation, which is the second derivative of the displacement with respect to time is equal to some constant, which is k by m times minus x. Okay, all right. But in a dynamical systems perspective, we usually write everything as coupled first order differential equations. Why? Because if you want to know how many independent dynamical variables are there describing the system, it equals the total number of first order differential equations that describe the system. So each of the variables which would be the variables described by the first order differential equations will give you a relevant variable of your system. Okay. So for example, here I can write it as two coupled first order differential equations. So first I write dx dt times m is equal to momentum, which I call p, and then the rate of change of momentum is equal to force, which I know is minus kx. So I can write one equation, one differential equation in the displacement x and one differential equation in the secondary variable momentum. So one of the variables is position, the other variable is momentum. So now if I plot the system in this space of the relevant variables which are described as first order differential equations. So along the horizontal axis I plot position, along the vertical axis I plot momentum. So then it's no longer a sinusoid, it won't no longer look like a sinusoid, but a closed curve. In general, it's an ellipse. Okay, the way I've drawn it such that the units I've chosen makes it look like a circle, but in general it's an ellipse because the units of position and the units of momentum are different. All right, and so what you will see is that in this space it will actually go round and round this closed curve. Okay, why? Because what is happening at this point here, the position or the displacement is 0. That means it's at its equilibrium position. At this point, the momentum is maximum. Okay, so it's either going this way or it's going backward. So depending upon whether it's here or here, the momentum is either positive or negative. It's either directed in this direction or it's directed in this direction. So depending upon how you define your positive direction, it will be either positive or negative. So you will see that it again and again goes through the x equal to 0 point. So that means it's once going through the equilibrium position, going towards the compressed position, and once it's going through the equilibrium position, going through the completely stretched position. Okay, what's happening here and here? Here the momentum is 0, here also the momentum is 0. Here the, what am I saying? Sorry, here the position was 0, here the position was 0. So here the momentum is maximum. Here and here the momentum is 0. Okay, so what's happening here? So when it reaches the state of the highest compression, for a moment it's static. So that means its momentum, its velocity is 0, that means its momentum is 0. And similarly, when it reaches its state of highest stretching, for a moment it's static before it starts its journey back. So that's when its position is maximum. So you'll have these 2 points, maximum displacement to the right and maximum displacement to the left. So you can basically relate each of the positions of the bob, which is located on the which is attached to the spring, as some point on this closed curve. Now, I can associate a phase angle with the position of this bob anywhere on this closed curve. So I can say that I can declare this to be my reference position, so this is my 0, and I can say that from here, wherever, I mean the instantaneous position of this oscillator at any point of time can be described by the angle it subtends to this axis. Okay. So, instead of talking in terms of position and momentum, I can now purely in terms of phase angle if I am not interested in the amplitude. Okay. So, in many cases, in almost all the cases we will see, we are not so interested in the amplitude of the oscillator, we are more interested in the phase of the oscillator. So, we will primarily be describing phase, and so where I will take this shorthand notation where I will completely ignore amplitude and only talk about the phase. So, you will see that I will be talking about d theta dt, where theta is the angle that is subtended by this point at any point of time with respect to this reference line. What's the advantage of this? Now I have reduced two coupled first order differential equations into just one differential equation, okay, namely d theta dt equal to omega if it's a homogeneous oscillator. So it's going around with a constant angular velocity, okay. And this is advantageous because I'll be talking about what happens when you couple many oscillators with each other in order to get waves, because that's essentially what we are getting at. We are not interested in individual oscillators, we are interested in waves. All right, everyone with me so far? If you have any questions, please feel free to ask me. Do you have any questions on this? Okay. All right. So, so one of the great benefits of doing dynamical systems is that you're not completely hung up on getting closed form solutions. So I don't care that, I know of course the solution has in terms of sines and causes, but I'm not really interested in the exact closed form solution. So I'll try to, I will try to do as much math as possible with pictures like this. Okay, so what's the relation between waves and oscillator? So normally the easiest way to show that is by using a wave machine like this kind. So I have my own, home built wave machine. So I need a volunteer. Can someone please come up and help me demonstrate the wave machine? Thank you. So can you hold that pink pencil end? Yeah, hold it tight. All right. So this is my home built wave machine. So what you have is a broad tape on which I have put a lot of colored pencils. All right. So each of these pencils, as you can see, is stuck. Okay. But they can oscillate. Okay. Right. And so what I'll be doing is I will be twisting them and letting them go. Now individually each of them is doing a periodic motion, of course extremely damped, right, because I'm holding this very tightly, so they'll be very quickly damped. But if I flick any one of them, you can see that there's a disturbance which is moving back and forth for a while, right. So this is the relation between oscillations and waves. Waves are phenomena that you will see in a system where you have coupled oscillators. Okay. So you can study various properties. Actually, it's not just a children's game. You can try to set up them in various different configurations, and you can try to see that if you use a very narrow tape versus if you use a broad tape, if you use very heavy pencils versus light pencils, what happens and so on. Okay. But for our purpose, the main thing I wanted to show you is that the pencils are all stuck. Pencils are not moving. Nevertheless, something is moving back and forth. So as you mentioned, is the disturbance. Well, I would put it slightly differently. I would say it's the configuration or some information, some information about the state of the system which is moving back and forth. Okay. Thank you. Okay. So this is a wonderful demonstration that waves do not require any material transport. Okay. It is some configuration or some information which is moving actually. Right? All right. So sometimes waves are very undesirable, right? So there's this famous case of the Tacoma Narrows Bridge in the US, which collapsed sometime in the early part of the 20th century. So there's a very interesting video in YouTube which shows how design flaws caused this bridge to actually resonate when the wind of a particular frequency was flowing across the narrows. Okay, so it's actually pretty dramatic. Let me see. Yeah. So this is a very old documentary. So this is, so you can see over there, right? It's starting to swing. So you can see that it has multiple modes of swinging. So there's one which is, along the direction of the bridge, and then one which is perpendicular. And okay, finally the amplitude has become so large that the strength of the material is not enough, and the whole thing collapses. Okay. All right. So this is a very spectacular demonstration of how waves can actually lead to fairly spectacular phenomena. Now let me go to something which is, a little more fun. Okay. Okay. It's a Mexican wave. Okay. So in a Mexican wave, what's happening is typically, you have these people kind of standing up suddenly, throwing up their hands, and then sitting down, and then the group next to them takes this up, and then the whole thing runs across the stadium. Okay. So again, another phenomena, the people are not going anywhere. They're just sitting in the seats, right? But something is traveling through the stadium. So what is traveling is this disturbance. Okay. So we could have a demo here if you are excited enough. So I have a fun video of this. As they say, don't try this in your classroom. So you can actually have a Mexican wave like this. Okay, so while this may look kind of funny, it turns out that this has a very serious analog in biology. So if you look at the embryo, okay, so the embryo starts its life as a single egg, a single cell, right? Upon fertilization, the cell divides, okay, into two, and then each of those two cells divide into two. So then from two you get four. Then each of those four cells divide into two, so you get 8 cells and so on. Okay. Now you got to do this in a coordinated manner such that you don't have one cell dividing again and again and all the other cells deciding not to divide. So then you'll get a very odd shaped embryo. Right? So how do you make sure that all the cells are dividing? Okay. You have to somehow synchronize them. Okay. And how do they divide? Each cell is actually guided in its division by a biological clock, what is called the cellular clock. Okay. So this involves a few proteins which are essentially controlling each other's expression. But for our purposes, we can just view it as a cellular clock. Every time that clock goes back to 0, you have a cell division happening. Right? Now, each individual cell can of course divide, according to this period, but that's not sufficient. You also need to somehow coordinate, as I said, their clocks. And this, it turns out, is done by these waves. And so this is an actual example of a embryo. So this is the embryo of your common fruit fly, Drosophila melanogaster. So each of these things that you see over here, the pink ones, those are the nucleus of a particular cell. Okay, and so what you will see is that as when I start the movie, so this is the anterior, that is the head, this is the tail, posterior, and when I start the movie, you will find that the cell division actually proceeds like a wave. Okay, so let me start it. Oh, sorry, I think it's starting from somewhere in the middle of the video. Okay, so I'll just wait until it goes to the end and then I'll restart it. So then it will be very clear what kind of, because by now already, you are having organization of the cells. So the cells are actually physically moving to form the, which part will become the, belly of the thing and which one will become the head and so on. So let me just replay this video. Yeah. So you notice, right? So every time there's this wave going through, the number of cells are doubling. Okay. So initially it's almost like you can't even see the wave. The wave is moving so fast. But then gradually the waves slow down and you can actually see a wave traveling from the posterior to the anterior. Okay. And by now it has reached enough number of cells so that it will actually try to segregate the cells to start forming the body. So this part will become the belly, this part will become the head and so on. Okay. Okay. So this is actually one very critical context in which synchronization or coordination of actions between various oscillators is set about by waves. So without these mitotic waves, the embryo will not know how to make sure that different parts of its body are growing in equal pace. Okay, so you'll have one enlarged head while the body is still small and so on. Okay, so the idea of synchronization is not new. So as far as we know, the first time someone wrote up about synchronization was when in 1665, Christian Huygens, this is the same Huygens who is responsible for the Huygens construction in optics, if you have done that. So like all scientists of his era, he was more than just being a physicist. So he would also build clocks. So clocks were a new innovation at that time, and it was very important for people to come up with as accurate a clock as possible. Now, it's not for keeping time at home that you need a very accurate clock. Why do you need an accurate clock? Where it becomes a life and death problem. Sorry? Navigation. Can you expand on that?
Back then, when they used to travel, they used to locate themselves by the latitude and time. So they needed time to figure out exactly where they were in terms of longitude.
Exactly. Very good. So latitude you can pretty much figure out using a sextant by looking at the position of the sun on the horizon at noon. But then okay, you might say, okay, to know noon also you might need a watch, but then you can pretty much say like, the sun only reaches up to this height. So you know that that's when noon is. So you don't, to get to know when is noon, you don't need really a watch. But longitude, it's ultra important to have a clock. Why? So because, you know when is noon, but in order to know how far you're away from, let's say, some standard longitude. So if you are using, for example, Greenwich meridian as your reference, so that means you're using the longitude which goes through the Greenwich near London as your 0 degree longitude. Okay, and so you need to know currently what is the time at Greenwich when it's noon wherever you are, to know how far you're located along the east-west axis. Okay, north-south axis you can get from the elevation of the sun, but there's no way of telling how far east-west you are without knowing the exact time at the reference longitude, which in most cases is Greenwich. But different countries would use different reference. For example, the French used to use Paris time as their reference. In fact, if any of you have read Tintin and read Rackham's Treasure, you know that one of the key plot points is that the original charts used in indicating where the treasure is located used Paris as the reference, whereas the calculations that Tintin and his friend Captain Haddock were doing were done using Greenwich time. Okay, so obviously the whole thing will shift if your reference itself shifts. Okay? So for navigational purposes, it was extremely important that you build a clock which would give you accurate time for the entire duration of your sea voyage, which can take a year. So you will lose less than a second, for example, when you're traveling for around a year. Now that requires extreme accuracy. So most early clocks would not be this accurate. It will keep losing time and so on, right, as the voyage progressed. So Huygens was also interested in this, and he was building a lot of pendulum clocks. And so in February 1665, he happened to be, sick with a viral fever, and he was lying in his bed, and he had two clocks that he was building which were on the wall. And quite idly, he observed that the pendula of these two clocks were essentially anti-phase, going anti-phase with respect to each other. So what is anti-phase? Anti-phase means when the pendulum of one clock was at its left extreme, the pendulum of the other clock was its right extreme. And if you let it go, it was going like this. Okay. Now, he was intrigued. He thought, okay, can this happen by pure chance? So he got up, then fixed the pendulums, and then let them go at arbitrary times. So no matter how many times he did this, after some time he saw that again and again it was doing this. Okay. So obviously whatever was causing them to come anti-phase with respect to each other, it was not just chance. Something was actually causing them to coordinate themselves in this anti-phase motion. So after a while, he realized that what's happening is each of these pendulum clocks basically set up vibrations on the wall on which they're hung, okay, because the wall is made of wood. And this vibration is basically coordinating the actions or anti-coordinating their action in this case, such that they were becoming anti-phase with respect to each other. So then he started experimenting by putting the clocks on these rods and putting the rods on these chairs. Actually, why would he do this? Why not just place them on the ground?
Different resonance of materials.
Yeah, actually, more to the point, see, you want the vibrations to actually interact with each other. If you put them on a surface like this, it will absorb all the vibrations. So actually, they will be decoupled. So that's why he wanted to make sure that they are interacting as much as possible. And by moving them with respect to each other, he could show that they could either go in phase like this, or they could go anti-phase like this. So he could do either of them. So if you are interested in doing these kinds of experiments for fun, actually there's a modern way of doing this. So for example, you could take metronomes. So these used to be very common when I was a physics undergrad. So we used to use metronomes for measuring various kinds of periodic behavior and so on. So actually, if you're learning music, well, I think digital metronome has become so common in cell phones nowadays that you don't use. But for example, people who would learn music in my time, they had to invest in these kinds of metronomes. So what you can do is you can see, you will notice that there's a weight. By changing the weight, you can actually change the frequency of the metronome. So once you let it start, it will actually tick, 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 tick at a particular frequency. So what you do is you take these different metronomes, you place them on a wooden platform, and these platforms are, themselves put on some soda cans. This is basically to ensure that they are on a platform which can move or vibrate, so that the vibrations are not absorbed by the ground. That's why it's not placed on the ground. Okay. Okay. So let me show you how this happens. So initially, someone goes and then sets them up at arbitrary phases. You deliberately, put them so that there's no relation between their phases. Now you see that there's small vibrations happening, right? So this small vibrations is what is causing this oscillators to interact with each other. Okay. So you wait for a while. Initially, you can see that they're all going their own merry ways. Okay. And in fact, even the frequencies are not exactly the same. The frequency is also slightly different. Okay. After some time, you see that it's starting to synchronize with each other. Okay. So here, these four have almost synchronized. This guy is a bit recalcitrant because it has a slightly different frequency, but then lo and behold, all of them are now marching in step. Okay. So essentially, just by having a coupling, which is through this mechanical vibration of the common platform, you can actually make things synchronize with respect to each other. Okay. So here also there is a wave, but what is happening is that once it has reached complete synchronization, the wave is actually instantaneous. Okay. The wave speed is effectively infinite. Okay. Okay. So if you are interested in this, I won't show this video because it's a pretty long video, but there's a very nice video made by this Veritasium, which actually has lots of nice videos on other subjects also. It's quite a long video which kind of looks at, the history of synchronization, how Huygens came up with this idea and then how it has developed further and so on. Okay. And there are also very nice demonstrations of, doing it with lots and lots of these oscillators. Okay. Now, why is this of interest to us? So, in my group, we actually look at biological oscillators. Now, you might say, okay, biological oscillators, what kind of things are they? Well, actually, you'll be surprised. There's lots and lots of phenomena in biology which can be viewed as oscillators. And they span a whole range of length and time scales. So, for example, brain activity. Okay, so if you measure, I'm sure you have heard of EEG, right? Electroencephalogram. So, if you go to any psychology department, they would have some kind of a EEG device. And nowadays, actually, these things can be bought off Amazon or something, where you'll get kind of a cap, shower cap like thing, which has lots of leads. So you can just put it on your head and it will record from the scalp the electrical activity, which is basically a attenuated version of what's happening in your brain. So your brain is of course operating electrically. The neurons are all emitting electrical signals. And this is aggregated at each point. And when you put an electrode over here, it's basically recording this aggregated activity. And so you can record from different points of your scalp and you would get, traces which look like this. So these waves have been studied for a very long time, so much so that each of these types of waves have a name. So for example, these are known as beta rhythms. So this is seen when you are awake, you're alert. Then you get this alpha rhythm when you are kind of sitting down quietly, not looking at anything. And then you got this theta rhythm, which is what you get when you get into deep meditative states. And finally, when you go to sleep, you'll get this typical delta waves. So in general, if you are studying psychology, for example, very standard experiment as part of your course would be to record these things. So we used to, in Calcutta University, the psychology department used to be in the same floor as the physics department and physics lab, and often they would take us as guinea pigs for their experiments. And so once it happened that someone nearly failed the psychology test because of a friend of mine who actually fell asleep while being recorded. And so instead of getting a beta rhythm, basically they got a delta rhythm, and the examiner was like, no, your machine is faulty. No, you don't know anything. And so the poor guy just slept. Okay, so that's one time scale. So this is actually very fast waves. Okay, so this is occurring at the scale of seconds. You could also have oscillations which are happening in your cells and which often lead to your circadian rhythm. So circadian rhythm is basically this body clock that we have. Okay, so even without an alarm clock, we wake up roughly at the right time, even if the curtains are drawn. How do we do that? Okay, because our bodies have an internal clocks. Okay, the enzymes, proteins that our cells produce typically have a period. This period is not exactly 24 hours. Okay. For different people, they are slightly different, but they are of the range of 26 to 27 hours and so on. And the reason why we actually are luckily, for example, synchronizing our activity with the sun is because the sun's 24-hour rhythm is basically forcing the slower oscillator to slave with it. So if you have a faster oscillator and a slower oscillator, you know that the faster oscillator will slave the slower oscillator. Okay, so that's what is happening. So because we are, if you let someone inside a room and don't give any cues, and if you keep doing this for days on end, you will eventually find that their sleep-wake cycle would start gradually going out of phase with the natural 24-hour cycle. So if you think that's a crazy experiment, well, this used to be very regular experiments done in the 1980s and 90s. So there was this very famous scientist at Madurai Kamaraj University who's one of the founders of the field called chronobiology. You know, how does biological phenomena keep time? And I've attended a couple of his talks when he was still active, and he mentioned that he used to run these experiments with people. Now, how do you get people to agree to be in a room for months without any cues? Sorry? Internship. Well, easier. PhD students, right? Sorry? Sorry, I didn't. Lockdown. Actually, yeah. So lockdown would have been fascinating if it was there in the 80s and 90s. This guy wouldn't have to even pay his PhD students, right? But yeah. So basically, he got his PhD students to isolate themselves. So basically, it would be like a completely closed room, and no clocks, only artificial light. So no windows, right? And food would not be given periodically. They will be asked to cook, because if you give food at regular times, then you can actually from that guess what is the periodicity. So you give enough food to them, and then occasionally you replenish it, but without the person realizing that you're replenishing it. So when the person is asleep, you like slip something in and so on. And the person is supposed to keep regular records of when the person is awake, when the person is asleep, and so on. And he found this pattern, which is of course he's not the only person to do these kinds of experiments. There were others who also did this. And there's a very natural setting in which this experiment actually happens. So when astronauts go into space, the spaceship in which they are there is actually rotating around the sun, or going around the sun at a period which is completely different from the 24-hour cycle, right? And so very soon they find that they are going completely out of phase with the 24-hour rhythm. Okay, so sometimes it happens that astronauts would work for, let's say, 30 hours straight and then sleep for maybe 14 hours or 16 hours and so on. So these kinds of very strange rhythms also happen. Okay. So so it used to be thought that the circadian rhythm is entirely based upon our brain rhythms, but now we know that even a single cell actually has a rhythmicity of its own. If you just look at what is the protein production on the cell, you will find that it has this regular periodicity. It reaches a peak and then reaches a trough over a period of roughly about 27 hours. So you know that there's a cellular basis for our body clocks. Okay. At other scales, locomotion gait pattern. So like when I walk, I have a particular rhythm of my own, right? So you can pretty much recognize that your friend is going around even if you can't see the face through the style of walking. So that means that there is an inherent rhythmicity to our walking patterns, but we are not conscious of it, right? So that's because the central pattern generators of our brain actually produces this rhythmic periodicity by which we kind of swing our hands and place our feet and so on. And we'll have different rhythmic patterns based upon whether we are running, whether we are walking, whether we are sprinting, and so on. Okay, not just us, even animals have very natural gait patterns. Okay, okay, and insulin release oscillation. So this is also very, very important, because if anything disrupts this periodicity, you will of course get diabetes of a particular type. Okay? So it turns out that insulin release happens because beta cells in our pancreas, the islets of Langerhans, are oscillating. And not only do they have to oscillate, they also have to synchronize their oscillation. So if they don't synchronize, the individual cells may be healthy, but they will not secrete any insulin. So this is the reason for several types of type 2 diabetes, for example. Okay, and then an even longer time scale cycle is the menstrual cycle. So this is basically over the course of an entire month almost. Okay, so all of these are oscillators located in our bodies, and they are, having very, very different time scales, from the scale of let's say seconds to the scale of months. Okay, you can even think of even longer time scales like annual cycles and so on. If you are talking about things like, for example, plants, okay, the flowering of plants and so on. So this will take place over an annual cycle. Okay, so biology is full of such oscillators, and often we find that it's not just the oscillators themselves that are interesting, but how the oscillators interact with each other. So one of the most spectacular example of how the oscillators interacting create a spectacular display is this example of male fireflies which are flashing in synchrony with each other, such that the tree on which they're located will light up all together almost like a Christmas tree. Okay? So this light that you see over here is just fireflies sitting on a tree, all flashing together. Okay? Now, if you're really interested in this, there's a very nice video clip which shows how this synchronization happens. So it doesn't happen all of a sudden. So if you go into the forests where this is seen, typically in Southeast Asia, and so this is actually shown in Trials of Life, which is a documentary made by David Attenborough in the episode Talking to Strangers, where he's shown on a boat going through this river in Southeast Asia. Initially in the evening, you see that these fireflies are flashing here and there, but they are not in synchrony. Okay, so you don't see any spectacular display like this. But as the evening proceeds to night, you see that one entire branch, all the fireflies have synchronized. Then gradually this synchrony spreads to the entire tree. And finally, almost an entire patch of forest will synchronize and light up. It's a spectacular display. Okay. Why do they do this? Yeah, to attract mates. Most things in biology is basically to attract mates. Okay. So, so one enterprising artist did this wonderful experiment. So what he did was, he took LED lights to the forest and he made this LED lights flash on and off according to a pre-programmed pattern. So he basically programmed it and then he set it to music, and so the LED lights will flash on and off, and he made the fireflies synchronize their flashing to this. So it's a very spectacular demonstration of how you can create a performance art not with humans but with fireflies. Okay, so you will see that there is some flashing of fireflies happening. So they are all uncoded. So this is a LED light. Okay, you can see from its steady rhythmic bursting. Okay, and so this is a firefly which is trying to synchronize with this LED. Okay, so then you can see that like using this LEDs, he's managed to make the fireflies synchronize themselves, okay, with this LED lights. So it may look like fun, but this actually gives us a handle as to how to analyze this activity. Okay. So what we can do is, we take this idea that we can use a periodic stimulus, okay, of a particular frequency, and we ask, okay, how does the firefly synchronize to this? So this model was built by two mathematicians, very famous mathematicians, Ermintrout and Rinzel, who have worked a lot on neural modeling. So they said, okay, let's take a description of oscillators which just uses the phase description. Okay, so I will not consider position momentum, I will just consider the phase angle. So that means that I've taken this line as the reference, 0 degree, and if your position of your oscillator in phase space is here, then the angle it makes with this reference line is the instantaneous phase, okay? And so d theta dt is equal to omega, which is the frequency of the oscillator. So different fireflies could have different speed at which they are flashing, so they might have different frequencies. But now imagine that you are giving this LED light stimulation at some frequency which is capital omega, okay? And since the flash is a very special event, we say that the theta equal to 0 corresponds to the flash. When the firefly flashes, small theta equal to 0, and when the LED light flashes, capital theta equal to 0. So that's the reference angle. Okay. So now how is the firefly flash responding to the stimulus? So without any coupling, d theta dt is simply equal to omega. So you need some function by which you need to express how the firefly is going to change its phase of flashing so as to synchronize with the LED light. So one possibility is you can use a sinusoidal function. Why sinusoid? Because or a sinusoid of the difference of the phases of the stimulus and the firefly's own phase. Why this particular form? Because what is known experimentally is that if the stimulus is running ahead of the firefly, so if the stimulus phase is ahead of the firefly flashing phase, then the firefly actually tries to flash early to catch up with the phase of the stimulus. So that means that if the difference of the stimulus phase minus the firefly phase is positive, then the frequency, the instantaneous frequency is increased by the firefly. On the other hand, if this phase of the stimulus is running behind, if it's lagging, then the firefly can actually delay its flashing. So you can say that if this difference is negative, then the angular frequency omega is actually slightly decreased to catch up with the stimulus. And so you can say, okay, it will be proportional to capital theta minus small theta. Why the sign? Any ideas? So what happens when the argument is small? When capital theta minus small theta is small, then I know that sine of capital theta minus small theta is almost same as capital theta minus small theta, right? So that part is easy. But why do I need a sign? What does a sign do? It bounds it over a range. So if you don't bound it, then you'll get this problem that very soon your d theta dt would be greater than the range plus minus pi. Okay? So you're going to get unphysical phase angles. Okay? So that's why you get this sinusoid. Okay? And what is this constant of proportionality A? It's basically telling you how strongly you're coupling the stimulus to the firefly's flashing. So if A is 0, that means the firefly is completely ignoring the flashing. Okay? But the bigger A is, that means the more the firefly is trying to change its phase to match the flashing of the stimulus. Okay? Alright. So now we can do a little bit of math. So we can say, okay, I'll just do a very simple replacement of variables. So I say that I'm interested in this phase difference, capital theta minus small theta. So let me represent it as some phi. And so what is the differential equation which describes how phi changes? It's simply the difference of the frequencies, capital omega and small omega. Remember, capital omega is the frequency of the LED light and small omega is the frequency of the firefly. So the difference of the frequencies plus A sine of the difference between capital theta minus small theta, which is simply phi. Okay. So just by substituting now, so you just make a small substitution. So you replace the difference of the two frequencies divided by this coupling strength as mu, and the time scale is also rescaled by this factor A, and then you get this very, very simple relation d phi dt equal to mu minus sine phi. Okay. Now, I will show you in a moment how to analyze this. Can you now tell me what you think? So here, which is the only parameter here? There's only one parameter in this equation. Mu. Phi is the variable. Mu is the parameter which you are tuning, right? So basically it contains two things: the difference between the frequencies of the firefly and this LED and the coupling. So depending upon, see, it basically tells you that if either the difference is small or the coupling is large, it will have similar response. Okay, so this is what you're controlling. Okay, so one parameter, one variable. What do you think the behavior of the system would be? How will it depend on mu? So if mu is 0, what do you think will happen? So mu equal to 0 means that either the two frequencies are the same, or A becomes so large, A tends to infinity. So it's a hugely strong coupling. So what do you think will happen if d phi dt equal to minus sine phi? What will happen? Sorry? What would be the asymptotic value of phi?
1.
1. D phi dt equal to minus sine phi. Don't try to solve it. Just again think in terms of qualitative dynamics. Okay?
Both of them will be in synchronicity.
Exactly. So basically phi equal to 0 would be a solution. And next we ask, is this a stable solution? That means if I make it phi plus delta phi, would delta phi go to 0 or not? Now you can try to do this with Jacobian and everything, or look at the slope, but this is a simpler way. You just plot picture. So what I'm going to do is I'm simply going to plot d phi dt versus phi, okay, and that is equal to sine phi, right? And so what this means is that here d phi dt is positive, here d phi dt is negative, okay. So that means here any flow would be directed towards 0, here any flow would be directed towards 0. So when mu equal to 0, phi equal to 0, that is the two phases would essentially converge. So I will exactly synchronize. So this will be the case if they have the same frequency or if the coupling strength is very, very strong. Okay. Now, if the frequencies are different and the coupling is intermediate, then you have, what do you have? So now you have a finite mu. So that's like, you are kind of slightly raising this curve bodily along the vertical axis. So now you notice it's no longer intersecting at 0. It's actually intersecting at this point. But again, this is a stable equilibrium. Why? Because the flow would be directed towards here on the left, flow would be directed towards here from the right from this unstable point. So you would be converging to a phase difference which is finite, but that phase difference will remain constant. So this is called phase locking. So the firefly is exactly flashing at a slightly constant time apart from when the LED is flashing. It's trying to synchronize, but it's always missing it by the same amount of time. Finally, if mu is greater than 1, that means the frequencies are very different or the coupling is very weak, then there is no intersection at all. That means the phase of the firefly is completely moving freely with respect to the LED. There's no synchrony. Okay? So this is called phase drift. Okay? So you can explain a lot of the what you are seeing in the field simply by using a very, very simple model of oscillator coupling with each other. Okay? Okay. Now, this is another example of a wave in natural setting. So this is where honeybees are on a hive and they're responding to a predator which is coming to attack them, like typically a wasp. So you notice that there is some kind of a wave which is propagating. But interestingly enough, you notice that it's not a simple circular wave. There is some kind of a curve. It almost looks like a spiral. Is this an illusion or is this real? Okay. So it turns out that it is actually a spiral wave, and there is a reason why spiral waves are quite common. It's not just honeybees which show this, but other kinds of systems also. Most importantly, it can be shown in chemical systems. Okay. So this, the first of the chemical systems which was capable of showing oscillations or periodic behavior, that is, they would periodically change in color, was proposed by Boris Belousov back in 1950s. So Belousov wanted to study Krebs cycle, which is one of the fundamental biochemical cycles in our body responsible for metabolism. And he actually set up an inorganic analog using cerium ions and bromate. Now, so this is the reaction. Don't worry if you don't understand. I don't understand it myself completely. I'm completely hopeless when it comes to chemistry. But what is interesting is that he saw that the state of the system can be represented by color as it went from a yellow color to a colorless solution as it went from a reduced form to an oxidized form. And the important part is that this color change occurred periodically. Unlike normal chemical reactions where you just see a color change and then the color change is static, this color change kept happening. Okay? So he wrote this up and submitted to a journal and it was promptly rejected. Why? Because second law says that once you have reached equilibrium, you cannot go away from it. And so people thought that, okay, color change is meaning that, it's somehow going to equilibrium and then again getting away from it. But actually, what is happening is that the system itself is very far from equilibrium. It's approaching equilibrium. So during that time, it's showing all this oscillation. So second law was fine, but nobody believed Belousov because they thought he was violating second law. And it went to his, he died. And then Anatol Zabotinsky, who was a young PhD student at Moscow State University, was given this problem by his PhD guide, mainly basically to get him off his hair. And Zabotinsky, basically his PhD guide told Zabotinsky, look, why don't you find out what's wrong with Belousov's reaction? And so Zabotinsky tried many things, and then he found out that no, it was perfectly right. And then he kind of came up with the solution, what's going on. So this is an actual experiment that we, sorry. It always happens when, let me see, yeah, so this is an actual experiment that we set up in IMSC. So we'll see the change in color. So this is basically the solution within a beaker. So you can see that it's turned from burgundy red to very light blue. And then you wait for a while, and then you will see it will again turn into blue. Then it turns to burgundy red again for a while. And then again turns blue, and so on. So you can pretty much set your clock to it. Okay? So if you actually record the concentration of the ions, you can see this oscillation, and you can see that the period is roughly the same. So what happens when you actually do it on a Petri dish, so that you can do it on a flat plane? You find that it actually generates waves. So this white is the front where it's actually showing this blue color, and this red is this burgundy red portion is the other part of the period. And you can try to describe this using so-called reaction diffusion equations. And you will see that here it's not just circular waves but also spirals, strange spiral shaped waves. And it turns out the spiral waves are actually all over nature. So it occurs in, for example, this amoeba called Dictyostelium discoideum when they're trying to aggregate under starvation conditions to form fruiting bodies. And you also see them in the heart. Okay? So in amoeba or slime mold, these spiral waves are basically a signal that they send to each other to ask other cells to come together so that they can form this fruiting body, and then they can form spores and then form the next generation. Now in the heart, we have this disturbances in the normal rhythm called arrhythmia. So this is an ECG taken from a patient who actually died. So this is the normal sinus rhythm, which all of you should have if you're in good health. But sometimes you get into this abnormally rapid heartbeat. So this is known as tachycardia. So tachy, as the name implies, it's very fast. So tachy means fast. And so you can see that it's beating 5 times compared to normal. And then finally you get into this fibrillation state where it shows no coherent pattern whatsoever. So at this point your heart has stopped beating, and if this continues for a few minutes, you are effectively brain dead. So what's happening during this time? So when people did imaging, so this is done over in a dog heart, you see that essentially this is because of a spiral wave of activity which is occurring in the heart surface. Okay, so this was actually first investigated by this Canadian physiologist called George Randolph Mines, who basically created a periodic ring by cutting the heart of a tortoise, and he showed that if you put it in solution, proper physiological solution, and if you stimulate an excitation wave, it will just go round and round and round forever. And this wave interferes with the natural rhythmic contraction of the heart, and it's interfering with it, and that's why it effectively will not allow heart to function properly. So you can model this using, not just the nonlinear dynamic part, but also a diffusive part which tells how the cells are basically interacting with each other. So instead of going into the math, let me just tell you what will happen. So the waves are propagating along the cells by traveling through these specialized proteins known as gap junctions. Now, these waves have a very special property. So normally when you have water waves or something which go through, which collide, they actually go through each other. But these excitation waves, when they meet head on, they actually just kill each other. Why? Because each of these waves basically have used up the resources behind it. And so it's like two fire fronts colliding. When you have fires coming this way and fire coming this way, when they meet, they will just destroy each other. Why? Because there's nothing left to burn behind the other wave. Okay? Exactly the same idea here. But here, now you have the possibility that you will actually create these kinds of spiral waves, and the spirals can themselves break and form this kind of multiple spiral waves. And so the reason why spiral waves form is that typically these waves can break, the open wave tip curls into this hook-like shape and then curls up into a spiral, and then this becomes unstable and then breaks up such that different parts are now, so wherever you have this deep red color, the corresponding cells are trying to contract, and basically you have no coordination in the heart at all. Different parts of the heart are trying to beat at different phases and frequencies, and so the heart effectively stops beating. Now, even at a larger scale, you can see spreading of epidemics in the form of waves. So this is an actual, SIR model. I'm sure all of you know about SIR models because of COVID. So this is basically, how measles spreads in Thailand. So you can see that it starts somewhere here. The red part is the infected portion, and then as the infection kind of starts spreading, it basically propagates down the narrow corridor of Thailand like a wave. Interestingly, if you look at how, for example, the Neolithic culture spread historically, you find that even that is like a wave. So this is where, the Neolithic, that is the change from a hunter-gatherer lifestyle to agricultural lifestyle happened, and that basically propagated as a wave. So you can pretty much date this. So this is from 6000 BC to 3000 BC. So you can see that with a roughly constant wave velocity, it propagated from the Middle East all the way to England. And there are other ideas, like the idea of a city. So urban civilization first started in the Middle East, and then gradually the idea spread all the way to England. So you can see that starting from 700 BC in Greece, by 400 AD, it reached England. Okay? So these are like, just a few examples to show how you can have, very interesting wave-like phenomena across scales, all the way from intracellular scales to population scales. You see waves, and all of them can be understood using the mathematical ideas that I very briefly sketched. So thank you for your attention, and I'm sorry to go a little over time. So this was my group a little while back. So Sridhar actually did a lot of work on this cardiac modeling. In fact, Sridhar and I have a book where we talk about this wave phenomena in the heart. And interestingly, it turns out that this wave phenomena also is seen, for example, in the uterus. So when a child is born, the uterus, which is for most of its lifetime is a completely quiescent organ, starts beating. The muscles become active. But the thing is that it's no use for muscles to be beating if they're completely uncoordinated. In order to push the baby out of the womb, they have to beat in a coordinated fashion. And so one of the puzzles is how do the myocytes, the muscle cells in the uterus, coordinate their action so as to push the child out. Okay? So this work was done by Rajiv, who actually tried to understand how exactly is this wave-like phenomena which causes the fetus to be pushed out of the womb happening in a coordinated fashion in the uterus. So if any of you are interested to learn more about this, I mean, we'd be happy to discuss with you. So we are at the institute, and I have several PhD students who are working on of these related problems. So please feel free to write to me or contact me if you are interested to learn more about this question. So thank you for your attention. I'd be happy to answer any questions.